And we're starting. All right. So I want to finish up. We just got started, but there's not a lot with the eukaryotic cell. And so I'll show you what is coming up next and soon. Um, so I just loaded this. This is basically this is the physiology of microbes principally, particularly bacteria and some eukaryotic microbes, kind of how they do business. And there's some really important issues. And I have a rather extensive outline there's a lot of useful lab information. And it's this one called Micro Chapter 4, even though the number is wrong because it's a different edition. But you will see it's a pretty, I mean, this is about pure culture techniques you're doing in lab and plating, but there's a lot about temperature and pH and water and required elements and some things. I mean, so it's a combination of stuff that you'll find useful for lab, I would anticipate. And probably the most important point of all of this is called we call it the bacterial growth curve, which is a lot of information from it. So I've outlined really a lot of the important stuff in the chapter for you. And so, and even though it's from a different text, I mean, they always cover the same things, just maybe a little different terminology. So that's there, as well as, let me click that, as well as, we may get a chance to start this today, hopefully, is the growth chapter, which is this chapter 6 from Callan. Okay, so I got that all over. Hey, it's good to be working, and we're still there and recording, and we're going to pick up where we left off in, that looks like chapter 4. Yeah. We really just started, but I'll frame it the way I did for the class this morning. And we're talking about, other than some specific stuff about microbes and their relevance, this is a eukaryotic cell. This is the cell we spent forever, literally weeks upon weeks in AP1 learning about. The idea of what's called understand for everybody going forward career-wise, if it's a prerequisite course, you're supposed to have a passing knowledge of that material. So it's not stuff that I'll try to go over and highlight it, but I got to go relatively quickly with the amount of stuff that we have to cover. So you're supposed to, I can't go back and tell you what's in the nucleus again in detail, or the endoplasmic and root, smooth and rough and bound and free ribosomes, blah. And believe me, if I'm going over it quickly, probably there's not a lot of information test-wise on it. There's probably a handful of questions, to be very honest with you. So, and I do have an older recording I'll probably post. I want to listen to it before I post it as we get ready. I'm thinking the 23rd, 2 weeks from today. Enjoy. You be back from track? Now we can definitely have it. You don't have to thank me. It's all part of the service. Throwing things at me will not help. So let's get on with it. So, oh, yeah, it's going to be like 65 questions, multiple choice, true, false, that deal. Stein's, matching once, twice, not at all. All those wonderful, you'll get used to it. It's not good. I don't sense this excitement. You look like the morning group. You got to stop staying up late. That was a lousy game. There was no reason to stay up. That was that, and they framed it with somebody I listened to this morning. That was like the only Super Bowl I think that I've missed was the first Steelers one, because I was on a plane to Vegas with a buddy of mine because we were on a break, whatever it was, early January or late December back in '74, whatever it was. I didn't have any doubt. But those original Super Bowls, there was typically a very dominant team. So despite my daughter-in-law, who I adore, who grew up outside of the Plymouth Rock in Massachusetts, sickening. Oh, where are the Patriots? So I had to tacitly say, "Oh, go Pats!" And of course, I'm suspicious because I've told you now that Tom Brady's technically available. My wife's very interested. It's okay. I can take the hit. He's got a lot of money. You're just awful. Okay, here we are. I talked about the flagella and the cilia. Okay. Okay, so we've got to look at all these different things. We talked about the membrane, right? Remember we talked about that, correct? Ergosterol, the whole. Yeah, yeah, yeah. I think we kind of were around the nucleus. Okay, by membrane-bound organelles, a standard nucleus. We talked about parallel membranes, the pores, DNA has to stay in, the other stuff, right? We did all that nonsense. Standard. Remember, it's going to be a test question. I'm sure of it. The nucleolus, where ribosomal RNA is made. I'm giving you a freebie. You don't have to thank me, money. And again, linear DNA. That's the big distinction. We have circular, and as you can see, when we get to mitochondria, and we talk a little bit about chloroplasts in plants and in algae, they have, and we're going to get to the endosymbiotic theory and why that's so important. So we have linear DNA in pairs. That's really important distinction. And it's chromatin because the chromosomes don't appear obviously until mitosis and meiosis. And understand, bacteria, as you will see in the next chapter, they basically just keep dividing in two with very, very rare exceptions. When you talk about eukarya, it's mostly mitosis for when we're dealing with microbes, mostly asexual. Some can do sexual, some can do both. Okay. Typically, it's more limited. Most of them are asexual, and some of them can basically, when you get to an amoeba, it's called fission. Okay, it's a little different than dividing into like bacteria, as you will see. It's like an amoeba will grow and grow, and all of a sudden, it'll burst, maybe the wrong term, but it will separate into multiple units. So mostly it's multiple fission rather than binary fission, though it's a similar process. Just to give you some context for why I'm going over all this stuff. And so we call it chromatin because it's associated with proteins that keep it in, where it is not supercoiled. But DNA is DNA. And you've seen these illustrations before. Go over rough endoplasmic, smooth endoplasmic, Golgi apparatus, etc., etc. All of those things that are there, it's fine. I mean, I'm not going to go, because we did it last time, the difference between free and bound ribosomes. And basically, I mean, this is kind of the way that a lot of our cells do business. We take a strand of messenger RNA, it's called a polysome or a polyribosome. And you have a whole bunch of ribosomes on the same strand. So it's doing multiple processing. It's like you have one template, and everybody here is making proteins from that. That's how, when you get to immunology, the immune section for many of you in AP2, that's how your body can ramp up and make antibodies in a matter of days in enormous amounts. And we're talking about cells making thousands and thousands of antibodies every few seconds. Just really ramps up. They're tiny molecules, but that's what fights many viral and bacterial infections, just for instance. So a lot of it is relevant here. Because most of you have AP2, I don't do the immunology unit here. I'll talk about it in passing and try to clarify it. I told this the other class today because I was discussing it with my stepson was over after he worked last night. We ate during halftime. Because he taught here in physics for quite a while as an adjunct. It's okay. You're allowed to ask questions. For whatever reason, there's a reluctance for people to ask questions in class. I'll answer your emails very promptly if you have questions and all that stuff. I understand that. Dialogues are sometimes helpful. It's okay. Just throwing it out there for future reference. I like talking to you, but I like talking with you as well. Yeah, it's all right. I could be one of those. We had those when I was sitting over there. Okay, Lou, here's question A. What is the, you go around the room and you get, I don't do that because I think that's not good management. I throw out generic questions and see if anybody's bold enough to answer. So here's the Golgi apparatus. We know it's processing and shipping and makes nothing. I'm not worried about transitional because they have CNs and TNs. Don't care. Okay, the assembly line. It's a nice way to phrase it. The endomembrane system. You might remember me making a big deal about that. All good. Nothing changed, and then they transport it out. Big deal here. Vacuoles it's interesting. Our cells don't have a lot of those. Microbes use a lot of them, again, for storage because they're small. And again, if they're trapped with air, they can provide buoyancy. Okay. Sometimes it's stuff that's been, been processed, it's been engulfed, all of those things. And again, and you'll have to go by and you'll see it here. We'll look at the typical endocytosis process because it's so important going forward. Again, from what your immune system does. And I'll just remind you of what we talked about last time. We basically talked about lysosomes last when we were in AP1. Yeah, it's a vesicle that contains very nasty enzymes for digestion and defense. Basically, when a cell dies, it releases these chemicals. So when you cut yourself, scrape yourself, those cells are releasing harsh enzymes that gobble up proteins, and it's why it hurts. And they stimulate, you'll find out, your immune system to come and visit you. Typically, it's a good thing, not necessarily a bad thing. But they're basically, that's sort of defensive. So basically, their role in digestion is anything you might remember, anything that's brought in by endocytosis brings in a vesicle. The lysosome fuses with it. Fancy name called a phagolysosome. And what it does, it releases those enzymes. And if it's nutrients or useful material, they gobble it up, break it down, store it till it's needed, and get rid of the waste. Sometimes you're going to see biologically, you'll see it in AP2 and digestive. Sometimes things are just encased in the membrane. They don't fuse with the lysosome, but they just transit through the cell. It's a process called transcytosis. You'll see that in the kidney. You'll see that in digestive. So they're important, digestion and defense. The other one that's pretty much all defensive in nature, they don't mention here, and I'm obligated to tell you about it because it becomes important in the next chapter. So it's called the peroxisome. I know I mentioned it, but I didn't go into the details, and I want to a little bit here. We'll go over the, not going to be a chemistry test, but I want to show you some things that are important, particularly relevant to microbes. So peroxisomes are mostly defensive enzymes, and what are they? You might remember what they defend. They defend against toxic derivatives of oxygen. I mean, oxygen is vital for a lot of organisms, obviously ourselves. But the problem with oxygen is what oxygen does for a living. Oxygen absorbs electrons. It's called the terminal electron acceptor, effectively bombarded with electrons. And anytime, even forms that don't utilize oxygen a lot still have to deal with it, and some can't. And that's going to show classifications in the next chapter. Ones that can deal with air, so ones that can't deal with air, one have to have it, ones that can't possibly get in touch with it. They're destroyed by it, interestingly enough. And so this is typically the bad boy. I'll draw brackets around it. It's called superoxide radical. It's a negative charge. It's unusual. It's an oxygen that picked up one electron rather than two electrons and splitting it in half. I know what happens. Free radicals bump into stable compounds and cause them to become damaged and create more free radicals that bump into this. You might remember the frame of it is that this is the modern source of cancer theory, modern source of aging theory. Things that alcohol, drugs, smoking, all are, we call them initiators and promoters. They start the damage and they continue the damage. Remember, I'm an authority, 40 years, 2 packs a day of smoking, that vibrant, active, went to the gym earlier last week. You're all looking at me. I'll show you my Planet Fitness app. Just itchy back there. And so what goes on with it, this is the way it works. I'm not going to do the chemistry. It's interesting. We typically utilize, in the presence of oxygen, most commonly, one of the enzymes that's there for defense is called superoxide dismutase. That's a mouthful, so we call it SOD. And it takes this and effectively, without doing the chemistry, typically in the presence of oxygen, changes it to this, H2O2. You know what that is? Anybody from chemistry? Hydrogen peroxide. Just like you go to the store and get the bottle. Hydrogen peroxide, which is also a free radical. That's why when we put hydrogen peroxide on a wound, those free radicals can be very nasty for any surface bacteria. Also, what happens on a wound when if you put hydrogen peroxide on intact skin, it doesn't do anything. It's there. If you put it on a wound, what happens? What does it make? Bubbles. It bubbles up. You're going to do that test in lab. We call it the bubble test. Because the other enzyme, so think of this as enzyme number 1, it takes peroxide, again in the presence of oxygen, and the other enzyme it has, enzyme number 2, is called catalase, and that converts hydrogen peroxide directly into water and oxygen gas. It's O2 with a little G next to it. So what happens when you put it on the skin, you have damaged cells. They've released from the peroxisome catalase. The catalase immediately contacts that. And what the catalase does is it rapidly changes, okay, the hydrogen peroxide, I beg your pardon, the hydrogen peroxide you put on the wound, the catalase rapidly changes, so you're hitting it with oxygen, and a lot of these germs, use a generic term, can't tolerate it, and the water doesn't harm you at all. So you're forcibly oxygenating them, as well as basically the rapidity of how intense that reaction is, speaking on a cellular level, gets rid of the bacteria as well as washing it out. So it's good stuff. Peroxide is nasty if you use it full strength. We're going to get to that. We do a whole unit after this one on antiseptics and disinfectants and sterilization, which is really the first really relevant clinical chapter, I think, and I'm very interested in it. So for instance, in the lab, you're going to get a gram-positive cocci. You're going to go, I wonder if it's staph or strep. The way we test it, staph has catalase. It'll bubble like crazy when you put peroxide on it. Streptococcus does not. So we call it the bubble test. It's one of the ways we test it. It's on the protocol. If I have my memory is good, I will show you a thing for solving unknowns. Tell them. I'm giving you help. And this is the whole phagocytosis, lysosomes, phagolysosomes, da-da-da-da-da. It's too small for my not so great eyesight to be able to see this. And so you can go over that. The big deal is not so much the structure of mitochondria, is to understand what mitochondria is. It's effectively what happened. The whole idea, maybe it's easier if I go forward than backwards. Endosymbiotic theory. The whole idea of what they now call this last common ancestor. We're talking billions of years ago. The first of our cells, by whatever scientific methods that were employed, and you notice archaea and bacteria predate us, was roughly 2.5 billion years ago. What was the planet? We didn't have an atmosphere. It was the harshest environment you can consider. We'll start with that. This last common ancestor now was thought to be a large organism with loose boundaries, with probably just RNA. And most likely viruses, individual cells, genetic material becomes DNA, possibly, notice the word. Whenever you see that in a book or in a PowerPoint, I've told you this before, permissive terms like that are short for I don't know or we don't know. So this is speculative, but scientifically speculative through the influence of viruses. The way HIV works, human immunodeficiency virus, which we're going to talk about at length. Okay, it has an enzyme called RNA reverse transcriptase. Basically, how it does its damage in our cells, HIV is an RNA virus, and when it enters one of our cells, the single most important white blood cell, as you will learn in our immune system, it rewrites it. So it rewrites the RNA into DNA. and it incorporates with the cell and takes it over and makes more little viruses, and we're in big trouble if you get in touch with that. Okay? So most likely this was a precursor of what we call a reverse. DNAs are called retroviruses because they go backwards. Transcription is DNA to RNA. If you reverse it, it's RNA to DNA. So they got their name retroviruses. I used to joke they would wear a leisure suit. Funny, go back historically. Watch old movies and disco. Leisure suit, ridiculous shirt, gold chains. We were like that. Even I was like that. Just saying. If you were only on East 86th Street in New York City in the summer of '74, you would have been in a disco. But I wager nobody was around. Nevertheless, then effectively now you have DNA, and probably what happened is this: most likely some kind of archaea, which was more likely to survive, rather than be infected by a bacteria per se. Endosymbiosis means this was invaded that cell, and rather than destroy it, effectively means to destroy it, incorporated it. That's the idea. And so what happens, that was probably the initial, the bacterial parasitize or engulfed by another descendants of that LCA, probably archaea. And the reason they say that, ribosomal RNA fragments and DNA fragments in us are much closer to the ones we found scientifically in archaea. So there's a lot of scientific for this endosymbiotic theory. They either become mitochondria or chloroplasts, which are energy factories in eukaryotic cells. Not all. Plants, ones that have chlorophyll, and chloroplasts rather, which have chlorophyll within them, also have mitochondria. They are the energy power plant, and we have plenty of our cells have, obviously, and a lot of prokaryotic cells have mitochondria for energy. And what they are most, and you're going to see the reason why it holds water is they have, as you will see, in the mitochondria, and the most, you don't have to know a lot about this LCA and who incorporated what and what it was. The two big deal things in our mitochondria. Here's the mitochondria. It has its own DNA, which is a single circle of DNA, which is prokaryotic bacterial type DNA, and it has 70S ribosomes. That's why you only see that in either mitochondria, or let's say in algae, you'll have it in mitochondria and chloroplasts. And that's so significant. And that's the big thing. Understand the two most important factors that substantiate endosymbiotic theory. And again, the word theory is significant because it means it's pretty well accepted by everybody. Is the presence of bacterial DNA, a circle of DNA, and 70S ribosomes. That's the test question, more or less, however I'll word it. Good? I'm making your life easy. I gotta stop doing that. So mitochondria, and they've got two membranes. More, I'm thinking gram-negative, maybe. Just a guess. And the cristae, and they have it, and this is aerobic respiration. They're oddball cells as well, but we don't want to get into it. And I had to make this point to the other class: do not throw things at me. I talked with some of the students who were in your seat last year, and apparently the nursing faculty, for those in that program, were a little surprised, probably the right word, that we didn't, whoever did micro, I only did one section, didn't talk about glycolysis or the Krebs cycle. I'm going to provide a little introduction. Has nothing to do with this test unit, but a little bit at some juncture in the next test unit. I used to do it all the time when I first started teaching. I didn't think it was that relevant because I was teaching mostly at Shady. I did all the micro for 5 years at Shadyside School of Nursing. And I was part-time. I was at Chatham. I was hired by Chatham University. But I was basically working with their students nearly, and sometimes shadow students. I didn't think it was that relevant, and the students didn't get it very easily. So I'm going to have to try to pick highlights for it. So I'm not going to teach the way I would do a bio major. I don't think you need it that much. If you're going into nursing, please keep my email. One day ask me if a patient ever asks you, could you describe the Krebs cycle to me? And I'm sure I will never hear from you. No one will ever ask you what you got in micro, or A and P. Okay. There's a one-word definition for people in healthcare, and one-word definition for nursing: caring. You care for them. It's like the famous military expression, the two-word definition for leadership: follow me. So all of those things, it divides independently of the cell. It's got its own DNA. Circular strands. It has bacterial-sized ribosomes. It can come and go. You go work out, your muscles make more mitochondria. Mitochondria have a lifespan. All of a sudden you get injured, you can't work out, you just got tired of it, and I'm going to break my New Year's resolution and whatever. And the next thing you know, they're going to disappear. Not because they disappear, because they have a lifespan, they go away. You just don't make more. So they come and go. The more active, if you're thinking more, you make more in your nerve cells. They'll show it to you with the illustration. Chloroplasts in algae, and particularly in plant cells, but algae are effectively microbes that are consistent with plants. They have a cell wall. They're the only ones that have chlorophyll or chloroplasts. They do photosynthesis. The good news is oxygen is a byproduct. You remember, and you're going to know that our metabolism is based on this equation. Glucose in the presence of oxygen gives us, okay, we're able to break that down, waste products of carbon dioxide and water, breathe out, get rid of moisture a number of ways, and a bunch of ATP. The reverse of that is photosynthesis. Basically, we take carbon dioxide and water, and in the presence of sunlight, which effectively is the energy input, when we have those chloroplasts, this is how they work. They take the sunlight and basically transform it into energy that can use, good news here, to make carbohydrates. I'll just put CHOs. And the waste product, you got to love this, oxygen. It's reverse. Glucose is a carbohydrate. You use oxygen and break it down for energy. That's life cycle 101. Got it? So you're going to hear a lot about that going forward. All that sort of energy type based type of stuff. So we have to, that's the beauty of having plants, having algae, the production of oxygen. Oxygen we can use and reverse the cycle when we need it. So they resemble mitochondria, the chloroplasts. They're larger. They contain the special pigments. Those are chlorophyll. When light hits it, it does energy boosting. It's kind of neat. In a funny way, it's kind of how lasers work, but I don't want to get into that. You do not want to know. I don't even want to remember that stuff. A long time ago. So ribosomes all over the place. We don't have to worry about here. They have both bound and free ribosomes. Bacteria just free. And again, they're normally ADS other than in the mitochondria or the chlorophyll. And the cytoskeleton, what's different, it's very elaborate. In our cells, the cytoskeleton is highly developed. We have the actin filaments. We learned about muscle. They play such an important role in that. They're the way they push to do endocytosis and exocytosis. They play a pivotal role in pushing out the membrane and closing it, opening it up. They can assemble and disassemble. They don't do that in muscle, but they do it all over. So we can build them and remove them. So they're so dynamic. They are the skinniest ones. The microtubules are the ones you saw with the 9+2. They're by diameter the largest ones. They're hollow. Okay, and the intermediate filaments are the ones that make the desmosomes, to hold the cell, tight junctions, etc., etc. Understand, bacteria, prokaryotic cells, have actin filaments only. They're called microfilaments. They're mostly made out of actin. They do play a role in the cell's shape and a little bit in its overall stability. They do not have intermediate filaments. They do not have microtubules. Those we only find in eukaryotic cells. And those actin filaments in prokarya probably don't don't assemble and disassemble in quite the same way or nor as frequently as in eukaryotic cells. So it has a different kind of, it's a much more elaborate side of it. I mean, we got all sorts of stuff going on in that cell. All these elaborate, there's just fibers and fibrous things and conduits and roadways and things like that within our cells are very busy and very or well organized. Bacteria, not so. They use their resources, I keep saying it, for reproduction. They don't need to, I call it elegant simplicity. They don't do a whole lot. This is a useful chart. And you can look through that. It gives you some ideas. Okay, bacteria are archaeal cells. They have a lot of the same stuff, but they don't have a nucleus. They don't have an envelope. They don't do mitosis, or for that matter, meiosis. The sex cell is very, very limited. I don't necessarily agree with that unless it's exotic. They replicate by binary fission. We'll get more into that. And they don't have all that neat structural stuff, as you can see. So it's pretty good. And you can look at the complexity here, all that stuff. And you look at the size as well. We'll get to viruses down the end. As you can see, viruses they have nucleic acids and a protein shell, period. That's it. And maybe a few other things. Some of them will have some external. We'll get into that. And so this is the endosymbiotic one. Great. Mitochondrion. Okay. A little bit here. So you'll see these in lab. Yeast, mold, fungi are all, they have a cell wall, so they're kind of like plants. Where algae live in the water and have chlorophyll, these are land and soil based. They don't have any, they're sort of like a hybrid because they basically do business like we do with regard to energy transformations and production. Okay, but they have a cell wall. So they're like an in-between thing. And what happens is they replicate kind of oddly would be probably the right way to say it. And they vary from the yeast phase, many of them, to the hyphae phase. And this is a big deal. These are called dimorphic structures. Two shapes. The ones that cause major problems in respiration are dimorphic fungi. It was the first yeast that really, when we defined the word AIDS back in 81, even though I will tell you, chapter and verse, it was around a lot earlier than that. It was because there was a cohort, a group of patients that were studied that had an unusual yeast mold dimorphic pneumonia called PCP pneumonia. And all of a sudden and the commonality was this virus, and that's where HIV was born and AIDS, et cetera, et cetera. We'll get to the story about that. It's not the only one. You hear about San Joaquin Valley fever, known as valley fever, histoplasmosis. We'll talk a little bit in respiratory about them. So yeast that are dimorphic can be problems significantly because they get these kind of strands. This is what they look like. You'll observe them in lab as they grow on a plate. They have it's not as, bacteria are pretty straightforward. You get the little dots. Sometimes they have funny looking colonies, but when you look at fungi, they grow interesting. They have different colors, different patterns, different shapes. Fungi I have a very extensive background in this. Okay. If you're like me and spent a quarter of a century treating people with lower extremity problems, you see a ton of athlete's foot, a ton of fungal infections. I'll tell you what I told the other class. Most people go, oh, that's just athlete's foot. Understand that damages a protective barrier, most important, your skin, and it allows for secondary bacterial infections. That's why you treat those things. If you don't treat them, it's laying the framework. And they comment a little bit more on that as well. Don't be dismissive of something because it's relatively common. merits treatment. Even if it's just simple to do at home. These replicate yeast. This is classic yeast by budding. Little buns form like that, and you'll see it. And if you take one of those little yeast packets, which you probably do in lab, I hope, and take a look at it, you'll see them right under there. Simple stain will show it to you. 300 species can cause disease. Environmental pathogens, I mean, they spread. Hospital associated, very nasty, and particularly what are called opportunistic infections. You're going to learn about what we call a compromised host. An opportunistic infection is something that would not affect typically you and I if we are healthy, but they affect people who are sick, immunocompromised, have cancer, have diabetes, which is an immunocompromised. And you're going to see a ton of it. I spent 25 years treating a ton of diabetic patients, vast majority of my practice. So I have a lot of experience with it. So that's the kind of stuff. And where do you see a whole bunch of these? In hospitals. The problem with hospitals, you got sick people. And they get sicker from things, and they're giving off these nasty. That's why you see the difference between hospital acquired and community acquired MRSA. They're much more difficult to treat. We classify them that way. I can't emphasize this enough. It is so difficult. That's why we do surgery mostly in outpatient centers. We don't keep people in the hospital. When I first admitted my first patient in the hospital in 1979, they went in the night before. They had the surgery there for 2 days. It's a relatively simple bone surgery. I broke a bone, reset it. They put a wire or a pin in it. That's all outpatient now. They come in the morning. They're gone by 3 o'clock in the afternoon. Most surgery centers, you can keep somebody there for 23 hours and 59 minutes. In an emergency room, you can do that because it's not a full day. Once a full day, now you have to admitting criteria. Most surgeons do not want to do surgery there unless they have to. There's higher rates of infection. This is what we're studying. That's because the bad guys are always there, and you have sick people that are basically culture plates. I hate to phrase it that way. For more bad guys. Okay, AIDS patients, substances, nasty. I talked a little bit about aspergillus. It's like this kind of a carpet. Probably they laid that down right on concrete. If it gets wet, aspergillus is everywhere. If it gets wet, and if you start to notice a smell from that, somebody in living, it was happening to one of my stepsons up at Slippery Rock. They rented an old apartment that was on the ground, like a chain of single level apartments. They were clearly on the cheap. When they moved in, they never replaced the carpet or anything. It was wet spots in the carpet, started to smell. I'm like, my wife and I went, you can get aspergillosis. That can be fatal. I mean, he didn't make friends with his landlord showing him pictures of that because the landlord, once he was notified, you have to go out and tear out those carpets. They didn't put padding into those things. Little stuff. Okay. So what they do to crops, very significant, very beneficial, all sorts of, we use so many of them. Antibiotics either come from mold and fungi and yeasts or from bacteria. That's their definition, as you will see. And we had a lot, obviously fermentation, different things, vitamins, and obviously we can ingest them. They're like us, they eat stuff that we do. That's what heterotrophic means. We talked about the fact that they were saprobes, they live on dead stuff. Okay. And parasitic is and typically we don't see them on, like athletes would be an example, on a living host. It's not necessarily as typical as you would think. And this is more lab stuff, their texture. This is how they replicate. Okay, and I'll show you because this is lab. And they even have spores, but they're not endospores. So they create endospore. I'll show you. It's easier for me to show it to you. This is something you'll look at in lab. You'll look at the reproductive structures, and that's how you categorize them. You have prepared slide in lab, and maybe you will grow some. Like if you have bread mold, typical white bread mold is penicillium. If you have strawberries, you got, and they kind of funky. I know that never happened to any of you. That's rhizopus, and you can prove that in the laboratory. And you probably will do that. So I don't go into all that stuff, nor do I know enough about the reproductive structures. or going into whether, and as you can see, they're either sexual or asexual. When I'm going through stuff fast like that, it's most likely something that's not going to be testable. That's why I do the study guide, so you'll be familiar with it. And then protozoans, like us, single-celled, no cell wall, water, and soil. So, so we can study them. There's a lot of different species. Most of the time, not a big deal. There are some. They are some of the most significant infection causers that we know. I'll give you two. The number one killer in sub-Saharan Africa, malaria, still. To from my reading, the second most, about 8 to 10 million cases in this country alone every year, new cases, ones that have been cured and come back, or people that have them, of sexually transmitted disease, okay? Trichomonas vaginalis. Much more numerous than chlamydia, much more numerous than gonorrhea. Ouch. That is protozoans. Thankfully, we can treat these. The malaria is a lot harder to treat. I'm not going to get into ecto or endoplasm or anything like that. Again, they move. We classify them how they move. Amoeba move just like our white. They just kind of scooch around. Like my great grandson I was showing you, they just scooch around and they love stuff, and they have cytoplasm, and it makes those pseudopods that are that. Somebody can have giant ones. They just eat like we do. They can scavenge, and we classify them a lot on how they move around in some of these special structures. And again, that's more lab than anything else. They can live in extreme environments, fresh and marine water, soil, and in us and on us. When we get to how, for instance, how chlamydia, when we get to that Trichomonas vaginalis, we'll talk about these stages. This is almost a dormant stage. It's a lot. When we see protozoan cysts, they're very hard to get rid of. Like general disinfectants, don't do very well for them. And you have this kind of thing you want to be wary about when you're cleaning up with, like, after, particularly if a dog was indiscreet, not that mine, inside the house, or you brought it in from outside, or the cat litter, the cats spread it all over when they're in a bad mood. Ours too. Did I tell you at one point we had 8 cats? My first one, she saw. As we say in Yiddish, a little mashuk. It's Yiddish for crazy. Nice person. Anybody who would marry me has to be a little mashuk. I'm only married for statistically 33 years. How long were you married? Well, I was happily married for 20 years. 20 out of 33 isn't bad. It's less than that. Dormant resting stage. This is the trophozoite, the bad guy. I don't happen to ask that question on a test. That's the kind of thing you see on licensure exams. What's the stage that's infective? Trophozoite, as you will see. And you'll see this again. Okay? And some of them are only in that. For instance, Trichomonas vaginalis is only in that. Does not form cysts. Okay? And it doesn't survive. Syphilis doesn't survive, even though that's a bacteria. Outside, they have to be treated. They are either intimate contact almost exclusively. Syphilis can be like from injections and bloodborne and things like that. These are ones that are very common. If you know somebody or you're familiar with somebody having well water, okay, and that means they have a septic tank, never the twain shall meet. So when you hear about giardiasis or amoebic dysentery, that comes from contamination in the water. Anybody who has well water has to be very aware of that. They all have something called reverse osmosis systems. So depending on where you live, where you work, be wary of that. Because it's typically, I know where I have lived around here has had even a boil water alert that I can recall myself. It doesn't mean they don't happen. They do. They had some problem with the ice by the water treatment plant down on the Allegheny by familiar where the Waterworks Mall area is, by St. Margaret's Hospital in Aspen. So, and again, relatively simple comparatively. There's some sexual. We classify them. They're very reproductive. This is a very interesting chart that gives you an idea of the diseases. And a lot of these are bad news, and we'll talk about some of these. Here, I mean, the intestinal, very significant. Malaria, vector-borne, comes from mosquitoes that are there. Toxoplasma, you'll get this from nursing. That's the reason why I had the 8 cats. You do pregnant women or women with, trying to be pregnant or are pregnant, have to stay away from the kitty litter because it kind of comes from cat droppings and cat urine and things like that. I had 8 cats. I was the sole changer of the kitty litter. Not going in. Don't want to talk about it. And we're not doing the worms. We are not get to the other chapter. Okay? So we're going to stop it here. I mean, the worms are interesting and egg laying, but I'm not going to get into it for our purposes. All right, everybody. I'm glad I finished that chapter. We will start with that chapter 6, which is kind of more or less the physiology. Thank you very much.